These mice are genetically identical. So why are they so different from each other?
Identical twins start out exactly the same genetically.
So why do they become so different biologically as they get older? Can what we do and what happens to us really affect the way our genes function?
And could our parents be passing on more than just their genes?
These are questions that are challenging the very basis of genetics. And they're taking us well beyond the age-old question of nature versus nurture.
Are you sick of your parents trying to control your life, giving you instructions about what you should do, how you should look, even who you should be?
Well, that's nothing. They were giving you instructions before you were born.
And genetics has taught us there's no getting away from these instructions, as they're in practically every cell of your body.
Wrapped up tightly inside the nucleus of your cells is your DNA, that contains most of the genetic instructions for making the proteins that make and maintain you. And these instructions come in duplicate, half from your mother and half from your father. So you see, they were on your case before you were even born. And it's these instructions that play a major part in making you, for example,
Tall,
not so tall,
dark,
fair,
easygoing.
or not.
The 20th century has been called the century of the gene. Technological innovations were giving access to life at the subcellular level, a micro-world never seen before, a factory of cells, molecules and proteins.
And by the second half of the 20th century, this research had given rise to a new model of gene function, sometimes called the central dogma. Genetic information is encoded in the cell's DNA. The DNA is transcribed into small, portable RNA messages. This is then translated to make proteins, the building blocks of life. And this process is called gene expression.
So, if you wanted to understand why a living thing works in the way it does and why it sometimes goes wrong, you had to track down the genes responsible.
And it seemed barely a week went by when you weren't being told researchers had done just that and tracked down the genes apparently causing or contributing to this or that disease.
And genetics went well beyond health and illness. There were genes that made us aggressive, obese, intelligent, or not, happy or sad. And apparently it was our genes that really decided who we'd fall in love with, or whether we'd fall in love at all.
And this idea filtered down to become part of our everyday culture. The old idea that your destiny's in the hands of fate or God was replaced by the new idea that it's really in your genes. But this view's starting to be questioned today.
For a long time we've had a concept of we can explain everything through our genes and we can almost justify things through our genes. It's not really my fault, it's in my genes. And we do inherit important genetic information from our parents. But I really don't think you can explain human complexity through genetics.
The idea that we are our genes, that we have inherited traits that effectively define us from our parents, is no longer considered to be the case.
And we can see both the expectations and the limits of genetics in the much heralded Human Genome Project at the end of the 20th century.
This was an international research project involving 2,500 scientists from 20 universities in 6 different countries to sequence all the 3 billion or so base pairs in the human genome.
The idea behind the Human Genome Project was that if you were able to understand the sequence of bases and the genes that comprise the human genome, you could start to make inroads into understanding how a variety of different diseases had the genetic underpinnings.
So hopes were high at the time. And the US President Bill Clinton, introducing the Genome Project, claimed it would revolutionise the diagnosis, prevention and treatment of most, if not all, disease.
But it didn't quite work out that way. The researchers were in for a few surprises. But it wasn't quite so much surprise, it was more Oh, that was a bit of a surprise.
And the first surprise was that we had far less functional genes than experts had originally thought. It was thought we had about 100,000 genes that code for protein, but in fact, we've only got about 25,000 at most.
And so one of the surprises was discovering that you couldn't really understand human complexity just by looking at the number of genes we have. And we're very similar to most other organisms when you look at those protein coding genes.
For example, human and chimp genomes are 96% the same, yet we don't behave like chimps. Well, not all the time.
Second, these active protein-producing genes only make up about 2 to 3% of our genome. We're not sure what the rest do. They're just bracketed away as non-coding or even junk DNA.
And third, sadly, sequencing the genome hasn't really revolutionised the treatment of disease in the way that President Clinton and so many others had hoped it would.
It's all very well understanding the sequence of letters that make up a gene, but that doesn't tell you what the gene does.
The genetic code's only the starting point. We've focused on it for far too long. It was great to know what the genome is. We need that information, but it's just a starting point. It's not something that defines all of human life or any other life on this planet.
Okay, so whilst everyone agrees that genes are really important and sequencing the human genome was a valuable contribution, it raised more questions than it answered, and some of these questions seem to be taking us beyond genetics, and it's that we'll be looking at next.
Lindsay and Tracy are monozygotic or identical twins, so genetically they're clones.
We were very, very similar when we were born. Tracy had a birthmark and I had a belly hernia, so that's how they kind of could tell the difference physically.
Identical twins have always been of interest to geneticists, and traditionally identical twins have been used to show the power of genes.
As students, you were given examples of identical twins separated at birth and brought up in very different environments. But lo and behold, when they were tracked down and reunited, they had the same hairstyles, they were doing the same jobs, married to spouses with the same name.
And there you have it, living proof that genes will out. Nurture? Forget it. DNA's your destiny. But there's another side to identical twins that tells a rather different story.
I think a lot of us are coming around to the idea of what's much more interesting is the way that identical twins are not the same.
Even though they've got identical genes, were brought up in the same household and treated the same way, there were some noticeable differences between Lindsay and Tracy, even early on.
Because you were more academic, I was probably a bit more sporty and a bit more kind of joining theatre groups and all that kind of thing. So we were starting to get different then when we got to secondary school.
Yeah, different interests and things. I was almost science-based, although like that, and you were more arty and creative.
And it's the same with most twins. Physical and behavioural differences tend to increase with age, and statistically there are significant health variations.
We know that if one twin has a disease such as schizophrenia, there's a 50% chance that their identical twin will also develop schizophrenia. To me, the interesting question is, why isn't it 100%? The very fact that it's only 50% shows you that genetics is not enough to explain this.
So there must be something else going on here besides genetics. And this is where epigenetics comes in.
This is a relatively new area of science that questions some long-held assumptions of genetics. And in particular, the idea of the central dogma we looked at earlier, that proteins are simply the final expression of the information encoded in DNA.
And epigenetics, meaning above genetics, questions this. And it's evolved into the study of biological process that changed gene expression, the phenotype, without changing the underlying DNA sequence, the genotype.
Epigenetics is concerned with how genes can be switched on and switched off, that is, they can be expressed or silenced, and how this information may also be passed on to the next generation.
These epigenetic instructions come from little chemical groups called epigenetic marks or tags that get added to the DNA and the proteins surrounding it. And this is how cells read genes. So, look at it this way. While the genome is like the hardware of your computer, the epigenome is more like the software, giving the instructions. And one way this epigenetic software functions is through DNA methylation, the addition or removal of a methyl group, which prevents gene transcription, effectively turning a gene off.
Another epigenetic process is histone modification. The long coils of DNA in our cells are wrapped around clusters of proteins called histones. How loosely or tightly they're wound round these histones controls how little or how much of the gene can be read and expressed.
So, while DNA methylation is more like a switch, turning genes off and on,
histone modification is more like a dimmer switch, allowing more or less of a gene to be expressed.
Distinct methylation and histone patterns exist in every cell, and this is what makes them different, in spite of their identical DNA.
We don't get teeth coming out of our eyeballs. Kidneys always stay kidneys. Liver always stays liver. You can't explain any of that through the genetic code, because if you sequence the DNA in all those situations, it will turn out to be the same.
A lot of this epigenetic information gets laid down during our embryonic development, but it continues throughout our lives. And how these processes work have important implications.
The differences in methylation, the differences in the way that genes are expressed, will have a bearing on cognition in the brain, for example, as well as susceptibility to a variety of different disorders and illnesses.
And the absolutely key thing here is that these epigenetic processes are responsive to environmental influences. So things such as food consumption, environmental toxins, stresses and so on can influence whether genes are on or off and how they're expressed. And this opens up a whole new way of looking at the age-old question of nature versus nurture.
Most scientists have known that genes and environment interact. What they didn't know was how they did it.
Now we're getting a much better handle on it. So we're understanding that a stimulus comes in from the environment. That could be cocaine or eating too much sugary food or being frightened. That changes signalling in our cells, and that in turn can lead to new epigenetic modifications being put onto particular genes.
And this helps explain why identical twins start to become increasingly different from each other.
Identical twins have identical genomes. However, the patterns of gene expression change as the identical twins age.
And this was shown from research in Madrid by Manel Estela. He studied 40 pairs of identical twins ranging from 6 to 66 and found significant epigenetic changes the older twins became, in spite of their identical DNA.
So now with epigenetics we have that link. We understand how the environment and the genes can interact together, and they do it via changes in epigenetic modifications.
So we looked at genetics and at the challenges presented by epigenetics. What we need now is an example. So we let Claire choose something close to her heart.
You've heard the old saying, you are what you eat, but maybe there's more to it than that. Could you also be what your mother, perhaps even your grandmother, ate?
Back in the 1980s, research had shown that the children of mothers who ate an unhealthy diet in pregnancy were at a significantly higher risk of cardiovascular disease, diabetes and obesity.
Trouble was, scientists had no way of explaining it. That is, until epigenetics, and a groundbreaking experiment with some now very famous mice. The year was 2003. The location, Duke University Medical Center, Durham, North Carolina. The researchers were Randy Jertel and his assistant, Rob Waterland. And the leading characters, some golden agouti mice. So called because they carry a gene called the agouti that makes them yellow, ravenous, obese, and prone to cancer and diabetes.
So, Jertel and Waterland wondered what would happen if they tried to alter this unfortunate genetic legacy, epigenetically. The idea, or hypothesis, was that maybe the food a mother eats is transmitted in her blood as chemical signals to her fetus, where it's then laid down as epigenetic marks on the fetus's own genes. So what they did was to put some of the agouti mothers to be on a health kick, giving them a diet full of nutrients such as vitamin B and zinc. And then they waited.
And what they found was that most of the baby mice born to the health club Great Diet mothers were very different. Brown, lean and active. They didn't become obese and lived much longer. But they still had the agouti gene. It had just been turned off by methylation from the diet.
And it's an amazing example of an environmental impact that actually gets transmitted to offspring and is epigenetic, not genetic.
So there you have it, nurture shaping nature. And while the agouti babies benefited from what their mothers were given, it can work the other way. Experiments exposing female or male mice to, for example, a poor diet, alcohol, cocaine, pesticides or stress found harmful effects transmitted to their offspring.
So if you're thinking, well, that's just mice, so what? Well, 99% of mouse genes have an equivalent in humans.
And so if something is happening in mice, there's a decent chance that it's probably happening in humans as well. It's just we may not notice it so easily in humans because of all the other stuff that's going on.
Of course, scientists can't do the same sort of experiments with humans. And epigenetics is still too new to have much transgenerational longitudinal data. But one thing you can do is look back to the past. And that's just what health specialist Lars-Olof Berggren did, using historical data from his hometown of Överkalix in the very north of Sweden on the edge of the Arctic Circle.
Back in the 19th century, Överkalix often became cut off from the rest of the country, and people living there were dependent on what they'd grown in the summer to get them through the long winter. And when the harvest had been poor, they practically starved.
But when it was good, and some years with the long summers it was very good, food was everywhere and every day was feast day. And Berggren wondered whether there were any differences between those who grew up in periods of feast or in virtual famine. So he compared a random sample of boys who experienced either feast or famine times when they were prepubescent, a crucial time for sperm development and laying down of epigenetic marks. And what he found was that the boys who experienced feast died on average 6 years younger than boys who experienced famine. But that's only part of the story.
Depending on whether they were undernourished or overnourished, that seemed to have an impact on their children and on their grandchildren in terms of how they responded to food, so such as their risk of obesity and diabetes.
When Berggren controlled for various socioeconomic factors, he found that the lifespan difference between the descendants was an incredible 32 years. So were these Överkalix grandfathers passing on more than just their genes?
Now, even to suggest this was absolute heresy in genetics, and Berggren's research paper was rejected by all the major journals.
And the reason that it's controversial is because it goes against all our standard models of Darwinian evolution and Mendelian inheritance.
Classic genetics tells us that while your behaviour will affect you and your health, it won't affect your genes. They're okay, they're safe, locked away, untouched for future generations. But since Berggren, epigenetics keeps finding data that's suggesting otherwise. Take this example from Bristol in the west of England. Data from a study of over 15,000 children born in the early 1990s showed that if their fathers had started smoking before puberty, sons were more likely to have significantly higher body mass indexes and therefore be more vulnerable to obesity and shorter lifespans, just as the Överkalix sons were. And these findings could have massive implications, not just for geneticists, but for all of us. It could be that the lifestyle choices you're making today will also affect the health and the life expectancy of the children you might have tomorrow.
I know. There's no getting away from it. There's a real downside to epigenetics. Something else to feel angry or guilty about. But there is an upside.
Epigenetics doesn't just control certain diseases. Epigenetics is also why we're mostly healthy most of the time. So it gives us great hope for understanding how we can keep ourselves healthy and not just sit around worrying about what genetics has given us in the great lottery of inheritance.
So first, there are things we can do for ourselves.
For example, while research has shown that obesity is linked to carrying copies of the FTO gene, regular physical activity can dampen its effect, so you don't have to become obese even if you get a bad throw of the genetic dice.
And second, epigenetics is creating new possibilities for what doctors can do for us. Scientists are developing new drugs aimed at reversing toxic epigenetic marks. For example, by turning tumour suppressant genes that have been switched off back on.
And we're already seeing the first generation of drugs that can control that process, and more work is being done to find new drugs that will treat even more forms of cancer.
But as exciting as these new developments are potentially, it's important to remember the jury's still out on epigenetics. True, for some it's a revolution, the biggest discovery in biology since the gene, while traditional geneticists are more sceptical. And as with most things, the truth is probably somewhere in between the two.
It tells us that there's a lot more flexibility in how the genome and the environment work together. We have to be careful though, it's like all exciting new fields, it's subject to quite a lot of hype, so we have to be careful about that.
I don't think there is any doubt amongst scientists today that the environment can alter the way in which genes are turned on and turned off. What is contentious is whether these changes can be passed to subsequent generations.
But it seems what we can be sure of is that the biological sciences are moving beyond genetics.
It takes us away from this tyranny of the genome where you're born with a particular set of genes and that's it.
So while 20th century genetics taught us that genes shape lives, 21st century epigenetics offers us another dimension, that lives also shape genes.
So yes, you do start out in life with the genetic script your parents gave you.
But it's the things that happen to you and the things that you do that will decide how that script is performed.
So it seems as important as they are, your genes are not your destiny.